Foi. Oi, gente, boa tarde. Meu nome é Giovana e eu vou ser a mediadora dessa palestra. Eu só queria avisar que a palestra vai ser em inglês, com o professor MYRON SPECTOR. Eu espero que vocês gostem. E se tiver alguém que entende, mas não fala muito bem e quiser mandar a pergunta em português, a gente dá um jeito de traduzir na hora, tá? Então pode mandar. Liga a chavinha para o inglês. Okay, here we go. Everyone, this is Professor MYRON. Again, thank you so much for being here. He's a professor of orthopedic surgery, Brigham and Women's Hospital, Harvard Medical School senior lecturer, Massachusetts Institute of Technology director, tissue engineering regenerative medicine lab, VA at Boston Healthcare System, and co-founder of the journal Biomedical Materials. He will talk to us about the advances in medical treatment enabled by biomaterials. Okay, so I'll be back at the end to read you the questions. Now it's all yours, Professor.
Thank you very much, Yvonne. It's a pleasure to be here. I wish I were there in person, though. I've never been to Sao Paulo, and I'm hoping to go someday, so we'll have to make that another time. You know, it's a pleasure to be here with you. I, too, am a biomedical engineer. My own work is in the subspecialty of biomaterials, so I'm looking forward to being able to chat with you today about some of the possible clinical problems that can be treated with biomaterials, and I hope you'll think about it for your own careers. It's been very rewarding for me, and I know you would enjoy it as well. So perhaps we can start with the slides that I have, that I brought. Good, thank you very much. So even though I'm in orthopedics, in fact, I'm not going to be talking to you today about an orthopedic problem. Instead, it'll be a neurological problem, and we'll get to this in a few minutes. Let me see if I can control the slides.
Cadê ele?
Tivemos um problema, ele deve ter caído, daqui a pouco ele tá voltando.
Ah, tá bom.
Um minutinho, gente. Gi, posso te deixar aqui, enquanto isso?
Pode, óbvio.
Tá bom.
Okay, sorry, let me get back to sharing the screen with you, if I can do so. I wasn't able to advance the slides. Let me see. Okay, 1 minute.
Don't worry.
Okay, here we are. Can we see the slides?
Yes.
Perfect. Okay, very good. So when we talk about biomedical engineering, you know, there's the biomedical piece of it and the engineering piece. And the biomedical piece involves investigating and beginning to understand the molecular and cellular and tissue responses to materials. But then we want to use the materials to treat a particular problem. So we need to begin to understand the degenerative processes, the effects of disease and injury and aging on tissues and organs that have caused the patient the problems. And then we would also like to know what wound healing may take place and if there's any potential for regeneration of the tissues and organs. So that's all the background. That's what we need to be learning when we look at the biomedical side of bioengineering. From the engineering point of view, you know, it's nice to be reminded that the engineering process is when we make a product using existing knowledge. This is distinguished from science, which is the process of acquiring new knowledge. And we want to use in engineering the engineering principles and practices of the various specialties in engineering, material science, chemical engineering, mechanical engineering, in order to produce these implants that might help treat patient problems and to analyze the function of the implants. Now, the reason for breaking this up is in this way is that as engineers, it's difficult to know everything, right? It's difficult to know all the biomedical aspects of the problem. But there's a message here in my presentation today. And the message is you can learn these. You can learn about the biomedical aspects of problems. It's quite interesting. It's rewarding. And it's actually often necessary for the engineer to learn about the biomedical features of a problem. You know, and the reason it's important to learn as much as you can is because, unfortunately, there is some bad engineering that goes in to make products that do not do well in patients. Sometimes that bad engineering is a result of a poor selection of the information and data that led to the design of the device. And of course, we do not want to be biased by any patents or proprietary know-how. We need to keep an open mind to select the most reliable data when we make a product, particularly a medical device, because a medical device can cause quite a bit of harm to patients. Even if it doesn't work, it can cause great harm. So we have to be especially careful in the medical device area. And of course, we want to understand what new knowledge is required. We never know everything that we would like to know when we proceed to design a medical device. And we want to be honest with ourselves about what we know and what we don't know. Because it may require, before we make the device, we have to act like a scientist and acquire that knowledge in order to make it the best device possible. When we talk about biomaterials, these can be either natural materials we take from the body or otherwise from nature, that take from the human body or otherwise from nature, or they can be synthetic materials used for the fabrication of medical devices. So that's simply what the definition of the word biomaterials is. Now, these materials may last the lifetime of the patient. They may be permanent in the body, non-degradable materials for the production of permanent implants, or they may be by design absorbable in the body to serve as an absorbable scaffold for the purpose of tissue engineering and regenerative medicine. And we're going to speak about one of those kinds of materials just a little bit later. Now, what I want also to get across to you, as I mentioned, there are some very interesting work that goes on in biomaterials, in making medical devices and designing medical devices. And when you look at this, you can be thinking, where would you make your contribution? What steps along the way interest you? Now, of course, this is a question that can be answered only after a period of time. You get experience, but it's worthwhile thinking about. So, for example, we start out before we make the material, we have to have a molecule of that material, and then we turn the molecule into a material, or we actually take tissues, could be from animals, and we process the tissues in which we create a new biomaterial as a natural biomaterial. And then from that point, we want to think about how to use the material to make the device. At this point, it's essential. We really want to be thinking about what the clinical problem is. How do we define the clinical problem? What are the specifications of the device that are necessary for that device to solve that clinical problem? And I'll be giving you an example as well in a few minutes. Then we need to evaluate that medical device in cell culture, in vitro, do mechanical testing on it. perhaps, and then implanted in animal models that may give us some idea of how that device would function in a human being. And then finally, we want to conduct a human trial. And if things are promising along the way, we want to think about getting into the clinic. And the way we do that is through commercialization. We start a company or have an existing company take on this project. And at this point, it introduces us to a lot of new activities, regulatory affairs, how are we going to get the Food and Drug Administration to approve the device, management of the people who are doing all the work, the financing, and the marketing and sales. So as you look at this slide, there are a lot of really interesting activities involved in commercializing medical devices. These are rewarding careers, and that you might find that you might want to think more about them for yourself. Now, I want to give you an example that we'll proceed more with, and that has to do with 3 organs that are part of the central nervous system. The stroke is a problem that occurs in the brain. Blindness, of course, is a part of a problem with our eyes, and paralysis that may be related to a spinal cord injury. And here we have some investigators with whom I've been associated working on these problems. And in all of these problems, we think a potential solution is an injectable gelatin-based gel that incorporates a particular kind of growth factor, epidermal growth factor. So the epidermal growth factor is in a liquid formulation of the gel. The gel as a liquid is injected either into the brain or into the retina or into the spinal cord. And once it gets into the body, it becomes a gel-like material that contains the growth factor, which then acts to stimulate certain cell functions. And these are the kinds of experiments and methodology used along the way. There might be a kind of rheology testing of the gels, how the gels flow, how quickly they set up, how stiff those gels become. Then, of course, we have to be sure that the cells that come in contact with the gel will not die. We want to make sure, of course, that the gels are not toxic. We also, of course, then also want to actually test it out in an animal model of the condition. So in this case, we induce strokes in rats, and then we inject the gel into the stroke lesion to determine if it's doing any good. Is it improving the function of the animals? And then at the termination of the animals, the sacrifice of the animals, we take the brains, in this case, and do histological evaluation. While the animals are alive, we do a behavioral evaluation, which I'll show you in a moment. Now, there are some similar experiments and methods when we also deal with using a gel for treating blindness or for treating paralysis. The reason I really want you to think about this slide is because of the investigators. These 4 young people were all students. They did this work as students. They had no prior medical background. In fact, some of them had not much prior biology background. And yet, look at the work that they did. They actually handled the animals, and they actually did the full experiment themselves, entirely themselves. So this I understand when you hear a presentation that's talking about stroke as a clinical problem or blindness as a clinical problem. You wonder, how could you ever make a contribution? How could you ever learn enough to do any experimental work in that area? And this is the message. The message in this slide is you can. You can. These are interesting as well as important problems. And certainly the motivation that you display, the motivation that these 4 people displayed, helped them get through this work. And The three of them obtained their PhDs doing this work. The fourth one, Pierre Colombe, is now about 6 months from getting his PhD. So I want to get that across to you, that this can be certainly complicated work. It's a formidable task, but you are all up to it. You are all up to it if you set your mind to it. Now, the job description, there's a reason to set your mind to it. These are good careers. It's very interesting to develop medical devices, working in biomaterials and developing medical devices. You save lives, you relieve suffering, you improve the quality of life of people. The career options are you could remain in university if you wanted to, or you can work in industry, or even be part of a startup company. And these is another list of some of the activities you could be doing. Investigating the synthesis and properties of new materials, fabricating those materials into medical devices, interacting with the clinical community. This is very interesting. Getting in touch, having discussions with doctors, surgeons to define the clinical problems that they have and try and figure out how one is going to engineer a device to treat the patient. Then it's a challenge to create the prototype medical device for testing in the laboratory and animal models. And then, of course, evaluating the tissue and organ response to and the clinical performance of these candidate implants. All these are very, very important. I didn't forget the income piece. It's plenty of money. You don't have to worry about the money to be made. There's plenty of money in this career, and as well as all these very interesting and important activities. Now, let me spend just a little bit of time giving you an example of how medical devices might work, how biomaterials may be used. And this is some work that we've done using an injectable gel-like material that incorporates a particular growth factor for the treatment of stroke. And as I mentioned, Chris Love and Texuan Lim were doctoral students at the time they conducted the work. Dr. Selim is a neurologist who himself treats stroke patients. Eng Lo is a neuroscientist, a very experienced neuroscientist. So we have a team. And it's very interesting when you do research as part of a team. You learn a lot from your other team members, and it's a very, very rewarding experience. So I want to show you Matt. This is Matt. And I want to show you because I like to start with the patient. I want to start with the clinical problem. I want to embody the clinical problem. This is Matt, and he looks healthy. He looks pretty healthy. He's a teenager, but unfortunately, he had a stroke.
Matt Gleason. Is that your name? Yes. How old are you? Yeah. 1, 5, 2, 17. Yes. And stroke. Football. You had a stroke during football? Yes. And
So I mean, it's sad, it's painful to watch this young man with this problem. He's had a stroke. There's a portion of his brain that's not functioning. So what is the expectation that we could ever treat Matt with a device, with a biomaterial that would enable him to improve in his function? So he would be able to better manage the words, better to think. So this is the goal. This is the goal. What can we do to help Matt in terms of a treatment? Now, looking at a young man like this, we have to get into the defect in his brain. So right off the bat, you know there's not a surgical procedure. You don't want to take a scalpel. You don't want to drill a big hole in the skull and take a scalpel through the brain, the otherwise healthy brain. So just by definition, it means that whatever we have to treat Matt with, whatever biomaterial, whatever device we have to treat him, we need to make it injectable. So we do not harm the healthy part of his brain. So when we think about injectable therapeutics, you know, we inject drugs. Drugs are small molecules. They can be anti-inflammatory molecules. They can be analgesics for pain relief. And this has been going on for hundreds of years. This is very old. This is very old and been very successful. I'd say rather more recently, in the past couple decades, in addition to injection of small molecules, there's an injection of biologics. Biologics are the products of living organisms or the organisms themselves. So the idea here is to improve the reparative process in tissues and to perhaps help facilitate regeneration by injecting, say, growth factors in cells. So this work has been underway. But I think what we're now seeing is a new wave, injectable biomaterials. Basically injecting a liquid that becomes a solid in the body, a solid gel in the body. And the reason we need this is because the defects are often fluid-filled cavities. So we want to provide the missing matrix or stroma in these fluid-filled defects. Now, what's of importance when we think about injectable biomaterials is that, as you see in the asterisk, these injectable biomaterials as treatments for various disorders will be necessary, but not sufficient. You'll need to have some kind of bioactive molecule, perhaps a growth factor, inside the gel in order to attract host cells into the matrix. So this is an important feature that we need to think about as we move forward, and I'll give you an example of. When we think about injectable biomaterials, the rationale is to deliver the therapeutic agent directly to the site at which it is needed. If we're treating stroke in a person like Matt, we have to inject the gel, we have to inject the growth factor right at the site at which it's needed. We can't give this to Matt systemically. We can't give it to him in a pill or put it in his bloodstream. We have to inject it into his brain. The injectable feature of the therapeutic approach is that it obviates the damage from surgical implantation. You just can't take a surgical blade into the brain to open up a corridor, a channel through which to implant some device. The injectable feature eliminates the side effects of systemic administration, and it also enables lower doses of bioactive molecules to be employed. Instead of delivering something subcutaneously or in the bloodstream directly, we can use much lower doses if we're putting the bioactive molecule directly at the site at which it's needed. Now, what's enabled this technology of injectability is that defects can now be clearly defined by imaging modalities. We can identify exactly where the lesion is, the stroke lesion is in Matt's brain. The injection site can be readily located by imaging. So we can locate all this, and also it's possible even to guide the needle as it goes into an organ using ultrasound as we do it, as the needle is being inserted. And now people are investigating, and there's more and more work using robotic devices for the injection of agents. So there's a lot of work going on right now to make the injection of a biomaterial into an organ like the brain, to make it doable. This can now be done. And also there's a lot of factors driving the development of these injectable biomaterials. It's a need to treat lesions which are irregularly shaped cavities contained within otherwise healthy tissues. That's the stroke lesion. In some cases, these can be very small defects. And these very small defects are the ones that occur in the retina of the eye that cause blindness. There's also a desire to employ cell therapy, use cells from outside the body, stem cells, for example. Let's use these for therapies. Let's use these to treat the patient. And many clinical trials are showing some promise. In all of these cases, though, the cells that are injected into the body of the patients, the cells are always in saline solution. And cells don't particularly like saline. The cells will perform better if injected in a more hospitable biomaterial matrix. In addition, there's the emergence of growth factors as well as drugs that require local administration, and controlled release is a way of delivering them to the site at which they're needed. And finally, there's a development all the time, there are new developments all the time of novel biomaterials that lend themselves to being injected. So there's a lot driving injectable biomaterials. What properties do we need of biomaterial matrices, these gels that could be injected? Well, degradation rate is important. They have to last long enough in the body before they're degraded to do their job. So they have to persist until sufficiently infiltrated with cells, the cells from the host, the cells from surrounding tissue, have to have enough time to migrate into the defect, migrate into the gel before the gel dissolves. That could be up to weeks. The gel that you inject, the biomaterial inject, has to flow nicely, has to fill the irregularly shaped defect. And when it becomes a solid in the body, when it undergoes gelation, it cannot be too fast or it'll clog the needle. And if it's too slow and just remains a liquid in the defect site, eventually it just may be absorbed into the body. So one needs a gelation rate that's matched to the particular application. Of course, it needs to have certain mechanical properties, like this modulus elasticity, and it has to have a nanostructure that's permissive of certain cell behaviors. Now, the asterisk you see here, when it comes to degradation rate, for example, or the mechanical behavior, in order to provide the best range of properties, requires covalent cross-linking to occur in vivo. As I mentioned, these biomaterials are actually liquids that are put into the syringe, injected through the needle into the organ. When they get into the body, they become a solid, but the solid is accomplished by having covalent cross-linking going on. It's the highest strength primary bond among molecules. So we want to be able to find the chemistry of the gel that enables covalent cross-linking to occur. If you just take a solid biomaterial outside the body, just take a sponge or something and grind it up into small particles, so the small particles go through a needle, that's not going to achieve the same effect. One needs to have a liquid formulation of a particular kind of polymer that undergoes covalent cross-linking in the body. So let's talk about brain injury. There's two types of strokes: hemorrhagic stroke, when you have bleeding, a blood vessel bursts and, excuse me, you have a blood clot formed in the brain, or the reverse can happen. The blood vessel gets clogged. It gets clogged, cuts off the blood supply to a portion of the brain, and that portion of the brain dies. That's ischemic stroke. And then there's another kind of brain injury, unfortunately, that occurs often as a result of, various kinds of injury, and that's traumatic brain injury. And you can see that these various types of brain injuries occur in millions and millions of people. They severely impair neurological functions. They're a leading cause of death and disability, and there is no current treatment for any of these. No current treatment. So that's what we're looking for, a treatment. So here's what they look like if you cut a brain in cross-section. As a result, say, of hemorrhagic stroke, this big area was filled with a blood clot. When the blood clot gets dissolved by the body, it's a process which is referred to as liquefactive necrosis. Basically, the body liquefies that blood clot, and you end up with a fluid-filled cavity, an empty cavity. There's no tissue there. In the case of ischemic stroke, you can also have various types of cells filling that stroke lesion, but these are non-functional. They're not neural cells. And you see that the gap here resulting from a traumatic brain injury. So these are challenging kinds of injuries, challenging kinds of lesions or defects to treat. And the treatment that we're investigating is a gel-like material made of gelatin. Gelatin is a degradation product, just you can get it from heating up collagen. Remember, collagen is a principal structural protein of your body. Your skin, the dermis of your skin is packed with collagen, right? They talk about pig skin. We talk about wearing leather shoes. Leather is just cross-linked dermis from animals, and that's all collagen. If you heat up collagen, you end up with individual molecules, chains that are called gelatin. If you take that gelatin molecule and link to it another molecule, something that looks a little bit like a particular amino acid, you enable the process of covalent cross-linking to occur. You see it occurs right across these near these hydroxyl bonds, as a result of the action of two agents. One is called horseradish peroxidase, this is HRP, and the other is peroxide. These are added, the HRP and added are added in very small amounts to the liquid formulation of the gelatin, HPA, and then within minutes or hours, you can control the gelation time of gel is formed, and you can also control how rigid that gel is by employing different concentrations of peroxide. Now, of importance here is there are many benefits of this particular kind of gel. Two of them are that one is that this gelatin displays cell adhesion ligands for neural stem and progenitor cells. That means these cells, which are so important to what we're trying to achieve, these cells can adhere to gelatin. They can adhere to gelatin. The other point that's important is eventually, after the gel does its job, we want it to disappear. We want it to break down, to degrade. And that degradation process can take place naturally because the same cells that we want to adhere to the gelatin gel, those same cells eventually release enzymes to break down the gelatin gel. So these are two of the important features that are offered by gelatin that have commended gelatin to us for this application. Now, there's a whole long list. There's a whole long list of properties that have been demonstrated by this particular gel that we're using. I mean, this work dates back almost 15 years. Many different studies done in cell culture and in various animal models have revealed that there's a lot of benefits on this particular gel. As I mentioned, the two of them, one is that the gel provides adhesion ligands for cells, and the other is that the gel undergoes enzymatic degradation. But there are many more advantages. The gel When you inject the liquid formulation, it becomes a gel in the body. The gel does not shrink and it does not swell. So there are, as I say, many benefits of the gel. Now, many of the benefits can be achieved only by incorporating a growth factor into the gel. So what's important about this is, as I mentioned earlier, for the applications that we're looking at, including treating stroke, this gel as a biomaterial is necessary to the treatment, but it is not sufficient. It just used alone, it's not going to work. We have to add growth factor into the gel before that liquid formulation is injected. This shows the importance of that liquid formulation. In vitro, one can do a little experiment to investigate how cells migrate into the gel. So on the right, you see this core area. This is the test gel. And on the outside, in this annular region, we've seeded another gel with cells. So this is like a tissue simulant. So we want to say that this area simulates the tissue with the cells in it. And we want to see how many cells will migrate into the gel. So that's the measurement that we're looking for. So here we are. We're going to look at these areas in these 7 panels. Are photographs taken of the gel, a core gel. This is the experimental gel that we're interested in. We put in here our gelatin HPA gel. And then here are the cells surrounding that gel. Now, if you look at 7 days after this experiment was set up, there are no cells, virtually no cells in the gel that does not have a growth factor. If, on the other hand, you have a growth factor, in this case, the growth factor is SDF1, that's stromal cell derived factor 1. If you put that growth factor in the gel, look at all the cells that come into the gel. And then you can adjust how you deliver the growth factor. Are you just delivering it alone, as in this panel, or are you incorporating that growth factor into a nanoparticle? So this kind of assay enables you to confirm that cells will migrate into your gel under the influence of the growth factor, and you can compare the effect of various growth factors. So here in the upper right panel, after day 7, if you have this growth factor SDF1, stromal cell-derived factor 1, in some nanoparticles, put them in nanoparticles, you get a lot of cells migrating into the gel. If you compare that to the blank gel, there's virtually no cells that have migrated into the gel. If you do an experiment in which you block the effect on the cells of this growth factor, you see how the number of cells drops, meaning the growth factor has been inactivated, so it's not able to attract the number of cells that it otherwise normally would. So it's proof of principle that a particular gel, when incorporating a growth factor, will attract the cells, but the growth factor alone, the gel alone will not attract the cells. The other important point is, when you run this experiment, if you add to the culture medium, if you add to the assay an agent that blocks enzymes, what happens is, if you block this particular enzyme, it's matrix metalloproteinase, MMP8I, if you block this one, in fact, the number of cells drops precipitously. It drops greatly. And the reason is that if you block the action of the enzymes released by the cells, those enzymes cannot contribute to breakdown of the gel, and the cells can't fit through the nanoporous openings in the gel. So it's really essential that the cells can degrade the gel ahead of them with their enzyme, in order to open up channels that'll facilitate their migration through this enlarged channel. Here's, you can watch the cells migrating into gel. Here's the core gel. Each one of these colored points is a cell, and the cell is migrating. You see the cell is migrating through the gel. The various cells, sometimes it migrates, comes back on itself. So here you can do several studies of what the cells are doing. In fact, you can actually track through a vector what direction they're going from. So the cells were first in this zone, and now they're migrating in these various directions. And the length of the arrow tells you how far they've migrated. So there's various experimental methodology that can enable you to better understand how the cells are going to migrate in your gel. Now, when we come to the animal studies, this is a typical frame that is actually placed on a patient when certain agents have to be injected into a brain of a patient, not necessarily for stroke patient, could be any patient. We're not talking about injecting biomaterials here, but this is a routine method for delivering drugs into the brains of the patients. And that same frame, we have a similar frame that we apply to the rats. Now, in the case of the rats, we induce a stroke to occur by injecting into the brain of the rats an enzyme, collagenase, which disrupts the basement membrane of the blood vessels. So we get bleeding, we get a blood clot forming. And we can adjust the location that we inject the collagenase, and we can also adjust the amount of collagenase we inject to cause a larger or smaller stroke lesion. Now, in a human case, if you look at the brains of patients who previously had a stroke, what you can see is that the kind of makeup of the lesion is generally a loose organization of cells that are not functional nerve cells. So that's the target we would have in thinking about the treatment in human patients. And we would be injecting this biomaterial-based therapy into the particular lesion. We can know exactly where the lesion is through neuroimaging like MRI, and we can guide the needle directly to the location of which it's needed. Now, in the rats, the protocol was we induce a stroke, an intercerebral hemorrhage, ICH. We induce a blood clot to form in the brain. And then 14 days later, we either inject our biomaterial gel or we inject a fluid, artificial cerebrospinal fluid, just a fluid that would be a control. This fluid is not going to form a gel. It's just a liquid injection as a control for the biomaterial that we're injecting. And then in the first of these studies, we sacrificed the animals 14 days after the gel was injected. Now, of importance here is that the investigator, at this time it was Tech Xuan Lin, the doctoral student, did a blinded evaluation. In other words, he evaluated behavior of the animals and also evaluated what their brains looked like under histological methods, but he did not know what group the animals were in. He was blinded to that. He had somebody else label the specimen, so he didn't know if he was treating an animal that was treated with the biomaterial gel or with just this control fluid. So the gel was injected into the lesion, as you see here. Once into the lesion, it underwent covalent cross-linking with time, and eventually it became a gel. So it goes in as a liquid, and eventually the modulus elasticity increases, demonstrating that the cell is becoming a solid gel. And this, for example, is 200 seconds. So we can adjust the gelation rate for the gel that we want. And this is what the cross sections of the brain look like 2 weeks after injection of the gel. That would be 4 weeks after the animals were received a stroke. Here is the brain, a cross section of brain in which only the control liquid was injected, artificial cerebrospinal fluid, and you see this rather large cavity. Now, in this case, what was injected 2 weeks after the stroke was induced is the gelatin gel containing the growth factor EGF, and here's the gelatin gel containing a growth factor SBF. And you can see in both these cases, the gel has filled that defect and is persistent. It remains 2 weeks after it was injected. Here's the gel alone, no growth factor. And here's the gel that contained the growth factor that had been previously incorporated into a particular kind of nanoparticle. Here's a little larger view of it. Here are 3 rats. Here's the gel in the rats. You see the gel filling this irregularly shaped defect in the rats. You see them here. Now, here again, a defect, a stroke lesion filled with the gel. This is 2 weeks after the gel was injected. Here's just the empty lesion. If we look at higher magnification, for example, in these 3 zones, 1, 2, 3, you see the gel. This is the gel here, very closely opposed to, almost attached to the brain tissue at these 3 locations. This is 1 here, this is 2, and 3. So you see also, I don't expect you to be aware of it, but when we show this to a pathologist and he looks at the cellular makeup around the gel, he does not see any inflammatory cells. So this demonstrates that the gel is biocompatible. It's not inducing any kind of inflammatory response. To show you how well the gel fills these irregular defects, here we have a measurement of the intimacy, of the closeness of approximation of the gel and the surrounding brain tissue. So you can see the gel has filled the tissue. Okay, there's a gap right here. There's a little gap here. There's just a little gap here, but for the most part, the gel in a liquid form has flowed into that defect, filled the defect, and then undergoes covalent cross-linking to form the gel. Here you see one brain that was injected with the gel, and you can see the cross sections cut through the brain. And at these various levels, you can see the gel in the defect. Now, there's a number of ways of doing behavioral testing on rats. We want to see if how the rats are doing. Without going these details, there's first is a modified, so-called modified neurological score. These are well accepted kinds of behavioral studies done on rats. And remember, when Tech Schwann did this evaluation, when he did the evaluation, he didn't know what group the rats were in. So what happens is normal is 0. Normal is 0. At this point, the rats received the stroke. So on day 3, after they got the stroke, you can see how abnormal their behavior is. That abnormal behavior in these 4 groups, there's 4 groups here, continues because it's only at 2 weeks that the gel is injected. Now look what happens after the gel is injected at 2 weeks. The gel that contains the EGF, gets better. That's the triangle. The gel that contains the EGF, that rat looks like it's getting better. The gel that contains the SDF, the stromal cell-derived factor, that growth factor, those rats look like they get better. The rats who were just injected with a control solution don't look like they're getting better. And the cells injected only with the rats injected only with the gel, not containing the growth factor, they may seem to get a little slightly better, but that's not statistically significant. The only statistical significant improvement are the rats that received the gel containing a growth factor. Here's another kind of interesting behavioral study that is routinely done on rats. You bring a rat close to a surface such that the whisker touches the surface, and then the rat will put his or her paw onto that surface. And what you do is you measure the 4-limb placing score, meaning normally rats 100% of the time they'll do this. They'll display this behavior 100% of the time. If, on the other hand, they get a stroke, you give them a stroke, they never display this behavior because they can't sense this and through their whiskers to know what to do. So here again, after a stroke, up through 2 weeks, the rats are still displaying this abnormal behavior. After injection of the gel, for the gel that contains the EGF, the triangle, or the square, the gel that contains either of these two growth factors, there is improvement. Now, they're not back to normal. They're not back to normal, but there's an improvement notably over the control that just received this, this control fluid, this control cerebrospinal fluid. Once again, the gel alone in the rats did not have the effect that the gel had when it contained the growth factor. And a third test was if you put a rat in a corner, half the time it turns left and half the time it turns right. So normally, if you measure the percentage of the turns toward the impaired side, the stroke side, you see that it's 50% of the time normally. Normally it's 50% of the time. They go 50% left, 50% right. But then after a stroke, for the 2-week period before they get the injection, they're not functioning normally. But once again, if you inject the animals with the gel containing one of these growth factors, in particular, one of these growth factors, you see they're starting to return to normal. They're not normal, but they're returning to normal. Whereas the other two groups, the group that had the gel only and the group that had this injection of the fluid only, the cerebrospinal fluid, they did not get any better. So these behavioral testing results demonstrate there's improvements in the rats early on, after 2 weeks after injection of the gel. And this early injection of the gel may reduce secondary loss of neurons. So it's not entirely clear what the mechanism by which the cells are getting better, but it provides some encouraging results for the gel. Now, if we look at the brains after the animals are sacrificed, what we want to see are red stained cells. So here are 3 groups. Here's the group, the control, cerebrospinal fluid in the defect. Here's the gel alone. Here's the gel plus EGF. So you clearly see these red markered cells, these red labeled cells. These are cells that have the potential to become neural cells. There are a fair number of them in the around the gel and migrating into the gel that was carrying EGF, but not in the gel alone, and not in the brains of the rats that received only the artificial cerebrospinal fluid. And you can quantify this. You can see the number of these nestin positive cells. Here's the number of nestin positive cells. If you look at the total number of nestin positive cells, the highest number occurs in the gelatin gel that's containing epidermal growth factor. So this is also encouraging results for that at higher magnification. Here's the gel. In this view, you don't see the gel structure. Here's the gel. Here are these nestin positive cells. They're sort of migrating, elongated as they're migrating into the gel. And of note is there's another agent that was given to the animals as a marker for the cell behavior. It's called Ki-67. And when you see these green dots in the otherwise red streaks, these green dots tell you that this particular cell, elongated cell that contains nestin, this cell is undergoing division. So the green dots show that the cells are not only migrating, but they're dividing as well as they migrate, which is a desirable process in order to give us more of these neural stem and progenitor cells. If you take one lesion and section it at various locations through the lesion, you begin to see if the lesion had been smaller, like this lesion or this one at the edge of the bigger defect, you can see that by 2 weeks after the injection, that entire lesion may have been closed. And there's other measurements that we can make of various cell types. Here's the quantification again of the neurals of these nestin positive cells. And there are other kinds of cells that we can stain for, including blood vessel cells. Here in this panel, the green shows that these cells are those that are capable of beginning to form blood vessels. Here's the edge of the lesion. Here's the portion of all this lesion is filled with gel. All of this portion, the peripheral portion of the gel-filled lesion, is now filled with these cells, and that process will proceed. And in fact, it proceeds through 10 weeks. Here are some 4-week results on the top. After 10 weeks in other rats, we see the process is still going on, and of importance is the gel persists in the defect. The gel persists in the defect, so it's there for the cells to continue migrating. And it's not surprising that this process of this reparative process in brains after stroke could take many, many months. Here's a higher magnification. Here's where the gel is, and you see these little dots are cells that have now migrated into this gel-filled defect. This is 10 weeks after that gel was injected. And you see here another image. These are just another image. I want to show you this, though. In this case, the right panel, the green stain is for the epidermal growth factor, that particular growth factor. And that epidermal growth factor here is showing up in gel that's been persistent in the rats 10 weeks. So it shows that the gel can incorporate a growth factor, but also importantly, retain it for this long a period of time. Well, I just wanted to give you some sense for the kind of work that can be done to demonstrate the potential value of various kinds of biomaterials, particularly those that are injectable, for treating formidable patient problems like stroke. So we're encouraged that this kind of treatment based on biomaterials, injectable biomaterials, may find a place in the clinic, and we're looking forward to continuing that. And we think that this is just the beginning of a wave of injectable biomaterials that can be used to treat these very challenging lesions, whether they're in the brain or the retina, the eye, or the spinal cord, for example. So, in conclusion, in addition to the students who did the work, we of course have a team of people supporting us, other investigators. The gel work that we've done is an outgrowth of work collaboration we have with an institute in Singapore. The retinal disease we have with another institution in Boston, and the stroke work we have is with our collaborators at Massachusetts General Hospital and also Beth Israel Deaconess. So thank you for your attention. It's been a pleasure to be able to to speak with you today. And I hope perhaps I've given you a little bit of an idea of the kinds of opportunities that are open for you in biomedical engineering. And I'm sure you'll find it to be a rewarding endeavor if you proceed along that way. So thank you. I wonder if you have any questions. I'll be happy to answer them. Please, Giovanna.
Thank you, Professor. The presentation was amazing. We do have a couple of questions. Okay, here we go. How long does the process of testing the device, etc., take? About how long? Because I know it's
No, very good. So it can take certainly a matter of years. It really depends upon how much prior work has been done. If you're starting from scratch, it takes a matter probably of about 5 years before you know whether or not you have something promising enough to think about commercializing. But it can vary. It can vary. Sure.
okay, perfect. What are the differences between an absorbable material and reabsorbable?
Good. You know what? Much of the time, those two words are used interchangeably. Same thing.
Okay, perfect. Next question. Are you working on COVID-19 related research?
No, I'm trying not to get COVID. I'm trying to stay away from it.
Aren't we all right?
No, but listen, I wish I knew something. I don't know anything about that field. So I'm a lay person like many others when it comes to.
Yeah, we just know we got to protect ourselves, right?
That's right.
Next question. What do you think about the precariousness and lack of medical treatment enabled by biomaterials in some countries, not only when it comes to the amount of people who receive this treatment, but also the infrastructure?
Yes, this is important. And I'm pleased to say that some of the former students who are participating in some of the gel work we're doing are very much focused in providing the treatments in what we might call sort of resource poor settings. I really am. So we don't want to make this expensive. And we do want to find a way to get it to people who need it, regardless of where they live on the globe. So that's exceedingly important. It can be done. And we're intent on doing as much toward that end as possible. It's very important.
Yes. Professor, a few days ago, we had a lecture about machine-brain interface used with quadriplegic patients. Could your gel have any application to restore these issues, either in the brain or spinal cord?
The answer is, I think, yes, it's definitely worth investigating. So, for example, in some cases, I think what will happen with time is when we see to what extent our gels can improve the function of a paralyzed patient, it may well be that improvement in function is not going to be perfect. It's not complete, but it may actually put some patients in a better position to utilize sort of a man-machine kind of interface or interaction. So absolutely. These different kinds of therapies may be able to be used together.
Perfect. One of your researches was about biomaterials and the challenges of aging epidemic. Could you please give us a brief about this paper?
Yeah. So this issue about the importance of treating the problems that occur with aging, of course, is exceedingly important. For me, as I get older, I think more about it as well. But we're also continuing to look at that, treating the the problems of aging in a way that is as little invasive as possible, right? I mean, fortunately, we're living longer. People are now fortunate to be living well into their 90s, but those elderly patients don't tolerate surgery so well anymore. So we're trying to find injectable solutions for their problems. But that too is very important problem to be dealing with.
For sure. Comparing the research in vitro and in vivo, are there more complications to overcome?
Yes. It turns out that one really has to do an in vivo study. It's necessary to do in vitro work when you have a lot of alternative approaches to try, and one can screen those approaches or screen variables in vitro. Absolutely, it's essential. But in order to really have an idea to what extent the treatment, perhaps the various biomaterial, will be of value, it is really essential to do in vivo study. And it's not easy. It's not easy, of course, but it's important to do so. And this is also, I have to say, where collaboration is very helpful. One can often find people who are doing work in vivo in useful animal models. They haven't thought about your approach, but it's worth talking with them. And often, ultimately, one can use a team-like approach using their animal model to test your material.
Perfect, thank you. Next one. How is the biomaterial gel managed for ischemic strokes, more precisely when there are dead cells occupying the area?
Very good. So what happens is there are two options. One is depending upon how porous a site it is in the brain, sometimes one can inject the gel directly into that site, even though they're dead cells. You leave the dead cells, you just inject the gel, and with time, what will happen is the dead cells will be removed by the body. The cells like macrophages will come and will remove those dead cells. So it's possible to inject the gel treatment even though there are dead cells. And that's something that we're working toward now to better understand.
Okay, next one. You said that the enzymes need to keep working on the gel so the cells can still grow. Was the gel totally absorbed in any of the test subjects?
Yeah, so what you see clearly is that as the cells migrate from the outside of the gel to the inside, they degrade the gel. There is no more gel at the outside. Wherever the cells are, there's no gel. So it's a synchronous replacement of gel. As the cells migrate in, they degrade the gel. So that's the process that appears to be going on.
Okay, we have one last question. Could you please comment about the status of applications of the nanomaterials technology in the injectable biomaterials?
I'm sorry, please say the beginning of that again.
Could you please comment about the status of applications of the nanomaterials?
Yeah, thank you, sure. Yeah, so what I've mentioned today, for example, what I mentioned today has to do with problems in the brain, or I mentioned the retina or a spinal cord, but there are many applications in other organ systems. So for example, one is dental. We're looking at the dental application. You know, you never think about it. When you go to a dentist, sometimes a dentist may see you have a little bit of loss of attachment to your tooth. You have a little periodontal defect. Well, we're working with a gel that contains an anti-inflammatory molecule, so the dentist could take just a little bit of the gel, inject it right at that site, and that would, in effect, reduce the inflammatory response and hopefully help stop that periodontal disease. So it could be a dental application, certainly in orthopedic applications. One can think about injecting the gel into various kinds of defects, perhaps into the intervertebral disc, for example. And then there are other applications to treat conditions where there are various types of problems. So one is a case of after surgery, some women develop adhesions in their uterus, post-surgical intrauterine adhesions. And there's a possibility that if the gynecologist after the surgery injects a little bit of the gel, then in fact there might prevent those adhesions from forming. So there are really a wide array of applications of these injectable gels, and we're looking forward to opportunities to sort of test them out, and others also. Listen, there's an increasing number of publications about injectable gels of various kinds for a wide array of applications, and that's very encouraging to see. There's a lot more work going on in this in labs throughout the world.
Yes, perfect. I think, okay, yeah, that was the last question. Professor, thank you so much for coming. It was such a pleasure to have you here. The presentation was amazing. You are amazing.
Thank you. Thank you very much. It was a pleasure. It was a pleasure. It was a pleasure to be here. All the best. All the best.
Thank you. Bye-bye.
Okay, bye-bye. Bye-bye. Bye-bye.
And happy.
Okay.
Thank you so much.
Yeah.
Ok, não, né? Ok. Gente, infelizmente, essa foi a última palestra da nossa 6ª semana de Engenharia Biomédica. Esperamos que vocês tenham gostado, aprendido várias coisas novas. Foram diversos temas e diversas palestras e palestrantes sensacionais. Nós, da comissão, ficamos muito felizes em preparar este evento para vocês. Queremos agradecer a presença e a interação de todos e agradecer principalmente a nossa universidade, PUC, e uma pessoa muito especial que fez a semana de todos os cursos ocorrer, a Gislene Borges. Então, obrigada, Gislene. E antes de nos despedir, vou chamar as pessoas que fizeram parte da comissão e tornaram tudo isso possível, né? Agora eles vão vir aqui dar uma mensagem. Primeiro, então, vamos chamar a curadoria, a equipe de curadoria. Eles vão aparecer já já. Acho que eles estão entrando.
Oi! Oi, gente! A gente gostaria de agradecer bastante.
Tem mais alguns. Bom, tudo bem. Se não chegarem, não tem problema. A gente representa eles. Curadoria. Não apareceu.
Começa falando, viu, enquanto a gente espera.
Até eles não chegarem.
Gente, a semana foi incrível, né? Então, eu gostaria de agradecer a todos e principalmente essas meninas aqui que estão na curadoria comigo, elas foram sensacionais. Tem algumas pessoas chegando, acho que a Gi caiu.
Voltei.
A Gi voltou. Então, eu só queria agradecer mesmo. Foi uma semana incrível. A gente foi atrás desses palestrantes e são palestrantes sensacionais que tornam a nossa SEB assim, inesquecível mesmo, né? Como nosso próprio lema dizia, co-criando uma SEB inesquecível, né? Então foi isso que a gente fez e obrigado mesmo, viu? Todas vocês e obrigado a todo mundo que ajudou. Obrigado mesmo, de coração.
Você, Vini, você foi incrível.
Não, foi incrível, desde a parte de mandar os e-mails, acho que pra gente que era do primeiro ano, foi uma experiência única, tenho certeza que a maioria das pessoas aqui vão continuar. E...
Gente, por favor, vocês têm que continuar, por favor.
Foi incrível conhecer e se aproximar de você, Vini, você é sensacional.
Obrigado.
Vocês todos, assim, veteranos, vocês aderiram a gente muito bem. Em vez de a gente se sentir mais parte da faculdade, né? Todo mundo. Então foi incrível.
Eu não estou emocionado, eu vou chorar.
Outra coisa, eu lembro que quando a Thalita falou, a SEB esse ano vai ser online, não sei o quê. Aí eu lembro que foi tipo, putz, que raiva vai ser online, queria ter feito ao vivo e tal. Mas eu acho que foi melhor ainda, a gente conseguiu trazer palestrante internacional, um monte de gente que eu não sei se poderiam ter ido se fosse ao vivo. Foi muito bom, aqui a gente gostou da situação não tão boa.
Mas, quer falar, Lai? Pode falar, desculpa.
Posso? Obrigada. Gente, para mim foi uma experiência incrível, ainda mais com vocês todos, com a Vi, com a Jana, enfim. Eu quero muito participar ano que vem, e se a Nicole e a Giovana tá aí, ano que vem também com a gente. Foi um prazer também poder estar trabalhando com o Vini, infelizmente. Eu não... Tomara que ele participe ano que vem.
Gente, vou tentar. Mas, sério, gente, obrigado de coração. Acho que a gente tem que dar oportunidade também para as outras equipes entrarem, para não pegar muito tempo.
Verdade.
Mas nosso time de curadoria sou eu, a Ni, a Gi, a Lai, a Vi e a Jana. Então, obrigado a todo mundo que está nessa equipe, e nas outras equipes também, né? Porque a gente fica se conectando e um ajudando o outro. Obrigado mesmo, gente.
Tchau, gente. Foi incrível esse evento.
Agora vamos ensinar o pessoal das artes. Da arte. Cadê vocês? Todos aqueles posts no Insta maravilhosos, com foto aqui, foto ali, foram tudo eles, tá? Que vão entrar agora.
Obrigado mesmo, gente.
Tchau, gente. Foi incrível.
Oi, tudo bem?
Cadê a Rafa?
Esperando a Rafinha. Pode falar, Rafa.
Oi, gente, eu sou a Rafa. Ai, calma. Aqui estão 3 pessoas da equipe de artes, faltam 2 que não conseguiram entrar, que é a Além e a Mandinha. Eu só queria agradecer por toda essa semana, foram 5 dias simplesmente incríveis, muito proveitosos, pelo menos na minha visão, né?
Foram mesmo. A gente aprendeu muito, é uma honra participar com vocês. Foi ótimo fazer parte disso, foi incrível, sério.
Perdão, a net tá horrível.
Tá valendo, Lu. E é isso, gente. Muito obrigada por terem feito parte desse evento. E nossa equipe, ela ficou, como a Gi já falou, ela ficou responsável pela arte de divulgação de toda a semana. Espero que vocês tenham curtido as artes, a gente fez com muito carinho. E é isso, gente.
Foi ótimo, gente. Amei, foi demais.
Obrigada, meninas.
Se fosse presencial, seria a hora que todo mundo aplaude, gente. Aí agora, pessoal de vídeo, eles que fizeram aqueles vídeos incríveis, maravilhosos, dormiram tarde pra caramba, né? E viraram à noite, mas fizeram um trabalho impecável. Então,
bom, aqui a gente falta, acho que o Vitor e a Sofia, que ainda não apareceram, mas a gente foi complicado até no começo, porque não tinha muito o que fazer, né? Porque faltava falar com o palestrante, tudo assim. Mas aí a gente se virou, organizou. Aí foi muito quebra-cabeça fazendo os vídeos, mas deu tudo certo. Acho que ficou legal, né? Não sei o que vocês acham. No meu terceiro ano fazendo a SEB, e para eles é a primeira vez. Espero que vocês tenham gostado também. E todo mundo que está assistindo também, espero que tenham gostado. Pode falar, gente.
Nossa, tá pior que o Mauro, meu. Tá pior que o Mauro.
Vitor, se você quiser falar um pouquinho, o que você achou?
Deixa eu ver. Espera aí que eu estou terminando de configurar aqui. Eu gostei bastante da experiência, tirando que o vídeo, para mim, foi o mais difícil de todos. Ter que renderizar, tanto problema que deu. Mas eu acho que deu para aprender bastante. Foi bom conhecer bastante gente. Gente que não conhecia da sala, por exemplo, por causa dessa quarentena, né? Deixa eu ver. Eu gostei bastante também que foi a primeira vez que eu participei assim, de uma comissão, de um evento que organiza alguma coisa grande assim mesmo. Aí eu me senti importante mesmo de participar de alguma coisa assim. Muito bom.
Se vocês quiserem falar essa experiência também, Ana, Natália.
Acho que foi isso também. Eu fiquei muito feliz de participar, deu tudo muito certo. Acho que foi muito gratificante de ver depois dessa semana um evento tão bonito, tão organizado. Acho que foi muito bonito mesmo de ver todo esse esforço que a gente deu desde o começo do ano, praticamente, desde que a gente se inscreveu, resultar nesse negócio tão bonito que ficou, tanta palestra bonita, e deu para aprender muito. Acho que foi muito gratificante por causa disso. Então aí, parabéns para todo mundo, né? Não só para a gente que fez os vídeos, que deu tudo muito certo, mas para todo mundo, que foi muito incrível, realmente.
Eu acho que é isso. Giovana, pode voltar.
E, por último, a equipe de infraestrutura, que acharam essa plataforma incrível, que deu tudo certo, que qualquer probleminha eles estavam lá para dar um jeito, que tiram e colocam a gente aqui. Parabéns para eles. Cadê?
Oiê, estamos chegando. Fred, vou te colocar.
Vou ficar devendo a fotinha.
Falta a Lê ainda. Acho que ela tá entrando. E tem a Tati hoje, que ela não pôde comparecer aqui no final. Também. Infelizmente. Vou ver aqui se a Lê consegue entrar de novo.
Pode me tirar.
Vou tirar um pouco você, Gi. Mas, gente, só queria agradecer mesmo. Obrigada por ter esses 5 anos de SEB, essa sexta, né? Eu tô participando há 5.
Essa é a minha terceira.
Foi
também.
A melhor SEB que eu já participei. A Lê chegou aqui, vou colocar ela. Isso. Pronto.
Oi, gente. Muito obrigada por todos que participaram, que ajudaram muito a gente. A gente teve uns probleminhas técnicos aí durante a SEB, mas deu tudo certo, isso que importa.
Sim, com certeza. Gente, muito obrigada a todos pela participação. Venham ano que vem, vai ser tão bom quanto, se não melhor. Esse pessoal que está aqui, o Fed, a Lê, que ainda vão continuar, vão arrasar demais. Todo mundo, todas as outras equipes, eles são muito, muito maravilhosos. Gente, muito obrigada mesmo. Eu agradeço de coração.
A gente tem que agradecer você, né, porque você comandou a gente, aprendeu tudo nas últimas semanas para fazer isso aqui funcionar e fazer da maneira que foi, como pegou todo mundo, esse pessoal todo assistindo, interagindo com a gente aprendendo a fazer cada vez melhor, e aí usar toda essa experiência que a gente pegou aqui e fazer as próximas SEBs serem ainda melhores.
Obrigada,
muito obrigado a todos.
Com certeza, Fred. Obrigada, gente. Vou colocar a Gi de volta.
Muito obrigada. E também a coordenação, né? Não sei se elas vão aparecer, mas Thalita e Carla, muito obrigada por tudo. Vocês vão aparecer ou não?
Elas vão sim, meu amor. Rapidinho.
A Gislene vai aparecer? Ela deve estar ocupada fazendo alguma coisa.
A Gi não, infelizmente. A Gi não, mas a gente vai esperar a Thalita e a Carla aqui, que elas falaram que iam conseguir entrar sim, mas a Gi, infelizmente, não vão poder. Pronto, chegaram. Pronto.
Lindo, lindo, maravilhoso, todos vocês maravilhosos. Queria deixar aqui público para todo mundo ouvir aí que...
Gente, não dá para ouvir vocês.
Chegamos.
Olá.
Eu acho que é orgulho, né, Carla? A gente está super orgulhosa deles.
Muito orgulho.
Eu acho que essa foi a melhor... Vocês estão ouvindo?
Sim.
Não?
Ué,
Nadine.
Oi, desculpa, gente.
Ele fala. Alô.
É só comentar, é só falar, gente. Ela tem um delay da live
então
daqui para o YouTube. Então vocês vão demorar um pouquinho para se ver, fiquem tranquilos.
Tá.
Mas podem comentar tudo que vocês acharam e tudo, que depois a gente tira, e depois vocês saem, tá tudo certo.
Tá, então, bom, eu acho que essa foi a melhor SEB de todas. Eu e a Carla, a gente está desde o começo, né, Carla?
Desde o começo, isso.
Bom, muito obrigada.
Eu só ouço a voz da Nadine aqui na orelha. E, bom, deixa eu falar então, eu queria agradecer aí toda a equipe.
Ô, Carla, eu acho que o problema é nosso, hein? Ó, os mais novos estão conseguindo, fizeram tudo direitinho, a gente está fazendo tudo errado, hein?
Eles ensaiaram, estudaram em casa, né?
É.
Mas eles estão de parabéns.
Bom, acho que agora a gente consegue falar. Não, Giovana, deu tudo errado.
Gente, muito obrigada a todos e a todas. Eu vou colocar o Enzo aqui de volta, rapidinho, que ele me pediu.
Eu queria agradecer. É a minha terceira edição, como eu falei, mas é a última da Nadine, a última do Vinícius e a última da Rafaela, porque eles vão se formar. Eu queria agradecer especialmente eles, porque eles foram que lideraram os outros grupos também, e não teria sido possível sem eles. É isso.
Muito obrigada, gente. A gente se vê na próxima SEB. Até!